All right, guys, this video is on DNA and RNA. So it's pretty straightforward stuff here. We always try to start off with some questions on these science videos. What are the four nitrogenous bases of DNA? We certainly want to know that. That's a common question. DNA replication, codons, what is messenger RNA, and what is transcription. So we're going to go over these concepts. And at the end of the video, these same questions will show up, and hopefully you can answer them. So let's get started here. So we start off with DNA. DNA is the hereditary material in humans and almost all other organisms, while genes are factors that control traits. Genes are the units of heredity and are a locus or a region of DNA. They determine what traits are expressed in individuals. DNA controls the cell's production of proteins. DNA is shaped as a double helix, which you can see here, which looks like a twisted ladder. Each rung of the ladder is made up of a pair of nitrogen bases. There are four bases, which are adenine, guanine, thymine, and cytosine. So you remember our first question? I believe it was our first question. What are the four nitrogenous bases of DNA? Again, adenine, guanine, thymine, and cytosine. So if you don't have that locked in your memory, we want to get it locked. Adenine always pairs with thymine, and guanine always pairs with cytosine. Also very important to know which bases pair with which bases. The sides of the ladder are made up of a pentose sugar and phosphate. This combination, a pentose sugar, a phosphate group, and the nitrogen base make a nucleotide. All right, let's go back to our first screen here for DNA replication, which is the biological process of producing two identical replicas of DNA from one original DNA molecule. This process occurs in all living organisms and is the basis for biological inheritance. DNA replication, second question, right? What is DNA replication? And then nucleotides, they bind together in specific patterns to form the genetic code that defines a given trait. Okay, and so that's all the stuff we really want on DNA. And so let's jump over double helix and into RNA. So RNA is essential in various biological roles in coding, decoding, regulation, and expression of genes. In order for a protein to be expressed, it first needs to be transcribed to messenger RNA. This mRNA is translated into protein by ribosomes. Messenger RNA is a large family of RNA molecules that convey genetic information from DNA to the ribosome, where they specify the amino acid sequence of the protein products of gene expression. And then we also want to know transfer RNA. So a transfer RNA is an adapter molecule, which is composed of RNA, typically 76 to 90 nucleotides in length, that serves as the physical link between the messenger RNA and the amino acid sequence of proteins. Okay, so there's everything we need to know about RNA. Go back to our questions. All right, we have the messenger RNA question there, the fourth one down. Okay, and then we'll jump in here quickly to codons, transcription, and translation. So a codon codes for amino acids. The amino acids are linked together as translation takes place. A group of 3 DNA letters makes up one codon. Transcription is the first of several steps of DNA-based gene expression, in which a particular segment of DNA is copied into RNA, especially messenger RNA, by the enzyme RNA polymerase. Both DNA and RNA are nucleic acids, which use base pairs of nucleotides as a complementary language. During transcription, a DNA sequence is read by an RNA polymerase, which produces a complementary anti-parallel RNA strand, which is called a primary transcript. And then on translation here, so in molecular biology and genetics, translation is the process in which ribosomes in the cytoplasm or endoplasmic reticulum synthesize proteins after the process of transcription of DNA to RNA in the cell's nucleus. The entire process is called gene expression. In translation, messenger RNA is decoded in a ribosome outside the nucleus to produce a specific amino acid chain or polypeptide. Okay, and that really gets us through everything that we need to know. So after having listened to me in this video, we should be able to answer these 5 questions that we saw at the beginning. So a great thing to do now, hit that pause button, see if you can answer these questions. If any of them are confusing, go back to the part of the video where we talked about it, and you should be good to go on DNA and RNA.